Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the DBT-JRF examination. So this is the day 19 of the free BET crash course in which I am going to discuss, as per your request, the most important topics from genetics portion, and in a very easy and in a very short period of time, I will explain some of the important topics which can be asked in this DBT-JRF examination as well as in the upcoming CSIR examination. So stay tuned and do complete watch the video and hit the like button if you like my efforts and hit the subscribe button if you want more from this channel. So we will start our today's video with this match the following type of format question. So in this A column, some of the important facts are given, and in this B column you have to match the exact function or the definition. It means the Turner syndrome. So what exactly is Turner syndrome? What is translocation? What is mosaic? What is polyploidy? So I will discuss all these terminologies in this single video, that what exactly is Turner syndrome, how it is caused, and translocation, mosaic, polyploidy, inversion, fragile sites, tetrasomy, translocation and nullisomy. In this video only we will discuss all these terms, and after completely watching this video you will be able to solve any question regarding all these terms. So stay tuned, and firstly we will see what exactly are these terms and then we will come to this slide to solve this question. I have included the answer of all these terminologies. So let's start with the first, Turner syndrome. So basically Turner syndrome is represented as the XO. The XO means the person suffering from the Turner syndrome has a X chromosome and the another chromosome is missing, so it is represented as XO. So the person having Turner syndromes are basically females, and if we talk about the frequency, it is only 1 in 3000 females. It means कि जो टर्नर सिंड्रोम है वो हर 3000 में से एक फीमेल को होने के चांसेस होते हैं, एंड it is particularly in females in which the secondary sexual characteristics are immature, like the menstruation or the menstrual cycle is usually absent, the breast development is slightly low, and some of the more symptoms like the pubic hair is very much low, and also the affected women are sterile and are frequently short in height and have a low hairline and relatively broad chest and folds of skin on the neck, and they are sterile. Now in the examination it will be asked that the Turner syndrome is represented as, so you can answer that 45. It has 45 chromosomes and it is represented as XO. And apart from this fact, Turner syndromes are basically females. You have to remember that they are females in which the one chromosome is absent, so therefore they have 45 chromosomes. Now if we have studied the Turner syndrome, we must study the Klinefelter syndrome, that is represented as the XXY mostly, but it can also be represented as the XXXY and the XXXY or XX and Y. So the person having Klinefelter syndromes with a frequency of about 1 in 1000 male birth. You have to focus on the key points. So the frequency is only 1 upon 1000 male birth, and the person suffering from this Klinefelter syndrome has one or more Y chromosomes and multiple X chromosome. It means they have one or more Y chromosome and they have multiple X chromosome. So basically a condition in males that can be represented as this XXY which is the most basic representation of the Klinefelter syndrome. But also it was observed that in Klinefelter males, the genotype is this XXY, this XXXY, or XX and double Y. So it can be represented as the mentioned genotype. Now 3 representation of the genotype of Klinefelter syndrome, and you have to remember that it is only 1 in 1000 male birth, and some of the symptoms include, though they are male, they have small testes, some breast enlargement, this is the recognizing feature of Klinefelter syndrome, and reduced facial and pubic hairs, and they are taller than normal and are sterile. I will provide the picture also of the Turner as well as the Klinefelter syndrome. So you can compare the visuals that how they look if a person is suffering from Turner syndrome or the Klinefelter syndrome. So basically you have to remember that in the Turner syndrome it is 45 chromosome and that is represented as XO, and in the Klinefelter syndrome it is represented as XXY, this one, and also the other 3. Now the third concept, that is translocation. So basically in a translocation, a chromosome segment moves from one chromosome to non-homologous chromosome or to another place on the same chromosome. So basically translocation में क्या होता है कि एक क्रोमोसोम का सेगमेंट, सपोज कि ये यहाँ पे जो एक क्रोमोसोम है ये सीक्वेंस है E एंड F, यानी कि एक जीन है E, एक जीन है F. नाउ उसी के होमोलोगस क्रोमोसोम में यहाँ पे है Q, यहाँ पे है R. सो बेसिकली ट्रांसलोकेशन में क्या होता है कि क्रोमोसोम का एक सेगमेंट या तो इट मूव्स फ्रॉम वन क्रोमोसोम टू द नॉन होमोलोगस क्रोमोसोम, या फिर एक ही क्रोमोसोम के दूसरे प्लेस में जाके वो एडजस्ट हो जाए. सो इन दिस फिगर यू कैन सी हियर दिस E and F. लेट मी क्लियर दिस फर्स्ट. सो यू कैन सी हियर इन दिस पिक्चर दिस E and F genes are exchanging. It means they are translocating onto the non-homologous chromosome. यानी E और F जो है वो Q and R से क्या हो रहे हैं, एक्सचेंज हो रहे हैं. QR यहाँ ऊपर चला जा रहा है एंड EF यहाँ पे नीचे आ जा रहा है. इट मीन्स क्रोमोसोमल सेगमेंट जो है मूव फ्रॉम वन क्रोमोसोम टू नॉन होमोलोगस क्रोमोसोम, एंड दिस इज नोन एज ट्रांसलोकेशन. इट मीन्स क्या हुआ है, क्रोमोसोम का सेगमेंट्स जो है उसका शफलिंग हुआ है फ्रॉम वन क्रोमोसोम टू द अदर नॉन होमोलोगस क्रोमोसोम. सो दिस इज नोन एज द ट्रांसलोकेशन. Now one more term from the genetics, that is mosaicism or mosaic formation. So basically you will be asked in the examination that what is the reason behind the mosaicism or the mosaic or the pattern formation or the patch formation. So basically the reason for the mosaic formation is the non-disjunction in a mitotic division. Now what is non-disjunction? So basically a non-disjunction refers to the failure of the homologous chromosomes or the sister chromatid to separate properly in meiosis or either mitosis. So it means कि जो homologous chromosomes है या जो sister chromatids हैं वो properly during mitosis और meiosis separate नहीं हो पाते हैं, and that is known as non-disjunction. So basically जो mosaic है it is formed due to the non-disjunction in a mitotic division which may generate patches of cell, and in every cell has a chromosomal abnormality. यानी कि जब भी non-disjunction होता है during a mitotic division, so it creates a patches of cell and every cell has a chromosome abnormality and other patches in which every cell has a normal karyotype. It means कि कुछ patches जो generate होते हैं उसमें chromosomal abnormality होता है हर cell में, and in the other patch every cell has a normal karyotype like normal person, And this type of non-disjunction leads to region of tissues with different chromosome constitution, a condition called mosaicism. And this condition, which type of condition? लाइक नॉन डिसजंक्शन से जो अलग अलग टाइप के पैचेस जनरेट हो रहे हैं, कुछ पैचेस में, कुछ सेल में क्रोमोसोम एबनॉर्मली हाई या लो हो जा रहे हैं, एंड कुछ पैचेस में जो कैरियोटाइप है वो नॉर्मल है, यानी नॉर्मल क्रोमोसोम है. सो द कंडीशन इन विच in some of the regions of the tissue with different chromosome constitution is known as mosaicism. And mosaicism is a very common condition in genetics. And if you talk about the Turner syndrome, जो अभी हमने पढ़ा, only about 50% of those diagnosed with Turner syndrome have 45 and X karyotype in all other cells, and most other are mosaics. यानी जिन पेशेंट्स में टर्नर सिंड्रोम डिटेक्ट होता है उनमें से 50% का ही सिर्फ कैरियोटाइप 45X होता है, बाकी सब में क्या होते हैं? ऑल आर मोजैक्स. इट मीन्स दे पोजेस 45X सेल्स एंड सम हैव 46XX सेल्स. इट मीन्स क्रोमोसोम एबनॉर्मेलिटी हो गया. हमने पढ़ा था टर्नर सिंड्रोम में कितना होता है? 45. इट मीन्स X क्रोमोसोम तो होता ही है. इट इज टर्नर सिंड्रोम वाज रिप्रेजेंटेड एज 45 क्रोमोसोम्स और XO, यानी ये नॉर्मल टर्नर सिंड्रोम का है. लेकिन कुछ टर्नर सिंड्रोम के पेशेंट्स में अगर मोजाइसिज्म होगा, सो क्या होगा? क्रोमोसोमल एबनॉर्मेलिटी होगा. सो यू कैन सी हियर कि 45X सेल्स के अलावा भी 46 और XX मिल रहे. इट मीन्स क्रोमोसोमल एबनॉर्मेलिटी हो गया. सो दिस कंडीशन इज नोन एज मोजाइसिज्म, and which is the result of or the consequence of the non-disjunction during the mitotic division or the meiotic division. Now a very important concept which is frequently asked, that is aneuploidy. So basically, in addition to the chromosomal rearrangement, chromosomal mutation also changes in number of chromosome. It means apart from the morphological changes in the chromosome, numerical changes can also occur, which is known as the aneuploidy. So variation in the chromosome can occur in two different types. That is, if changes is in the number of individual chromosomes, that is known as aneuploidy. And if it is changes in the number of chromosomal sets, then it is known as polyploidy. यानी इंडिविजुअल क्रोमोसोम में जब चेंज आता है, कम या ज़्यादा होता है, that is termed as aneuploidy. And if the whole chromosomal set is either increased or decreased, then it is termed as polyploidy. Now, please take a screenshot of this slide or either listen carefully or make a notes in your notebook that what is nullisomy, what is monosomy, what is trisomy, and what is tetrasomy. So basically, if you are talking about nullisomy, remember the formula 2n minus 2. If you are talking about the monosomy, remember 2n minus 1. And if you are talking about the trisomy, it is 2n plus 1. And if you talk about the tetrasomy, it is 2n plus 2. So the nullisomy, as the name represents null. Null means 0. So the loss of both members of a homologous pair of chromosomes, and it is represented as 2n minus 2, where n refers to the haploid number of chromosomes. So 2 into 23 minus 2, it means 46 minus 2 is equals to 44. So person suffering from nullisomy has only 44 chromosomes. So you have to remember that in nullisomy, 2n minus 2. Now monosomy. Mono means single. So a loss of single chromosome that can be represented as 2n minus 1. So 2 into 23 minus 1, it means 46 minus 1 is equals to 45. So person suffering from monosomy will have 45 chromosomes. Now, person suffering from trisomy, it means a gain of single chromosome, that is 2n plus 1. It means 2 into 23 plus 1 is equals to 47. So, a person suffering from trisomy will have 47 chromosome. Now, similarly, tetrasomy, it means 2n plus 2. Now, 2 into 23 plus 2, it means 46 plus 2, 48 chromosomes. So, tetrasomy person will have 48 chromosomes in total. So, you have to remember that in nullisomy, it is 44. Monosomy, it is 45. Trisomy, it is 47. 7, and in tetrasomy it is 48. So this can be asked in either match the following question or either in single format question. So you have to remember that, and also the formula can be asked in the matching format. So remember it, and you can also take the screenshot. Now after the aneuploidy, the polyploidy. So most eukaryotic organisms are diploid. We know that is 2n. And diploid means 2 sets of chromosome. Occasionally the whole set of chromosome fail to separate in meiosis or mitosis, which leads to polyploidy. We have just now read that when a changes in the whole chromosome set, either increase or decrease in the whole chromosome set, it is known as the polyploidy. And when there is a change, numerical change in the individual chromosome, then it is termed as aneuploidy. So the presence of more than 2 genomic sets of chromosome is termed as polyploidy. So polyploidy includes the triploids, that is 3n, tetraploids and pentaploids. Now I have also included the terminology that is non-disjunction. So non-disjunction refers to the failure of homologous chromosome or sister chromatid to separate in meiosis or mitosis. यानी अभी जस्ट हमने देखा था नॉन डिसजंक्शन, यानी कि प्रॉपरली जो क्रोमोसोम के होमोलोगस क्रोमोसोम्स होते हैं और सिस्टर क्रोमैटिड होते हैं ड्यूरिंग द माइटोसिस और मियोसिस प्रोसेस, वो पूरी तरह से सेपरेट नहीं हो पाते हैं, एंड दिस रिजल्ट्स इन मोज़ेसिस, अभी हमने पढ़ा था. नाउ वन मोर टर्मिनोलॉजी दैट इज फ्रेजाइल साइट्स. सो बेसिकली फ्रेजाइल साइट्स आर क्रोमोसोमल रीजन्स which are susceptible to breakage under certain conditions. It means fragile sites are constrictions or gap which offers a susceptible breakage chances and which are prone to breakage under certain condition. यानी fragile sites कुछ chromosomal region में दिखते हैं, होते हैं, and they are very much prone to break. Now come back to this match the following question. आई होप अब आप इसका आंसर दे पाएंगे. सो बेसिकली टर्नर सिंड्रोम रेफर्स टू दी 45X, दैट इज XO. इट हैज़ 45 क्रोमोसोम एंड द 46th क्रोमोसोम इज मिसिंग. एंड ट्रांसलोकेशन refers to the alternate segregation of the chromosomal segment we have seen. Now mosaic refers to the mitotic non-disjunction. We have read that it is the consequence of mitotic non-disjunction, that is the improper segregation of the chromosome, homologous chromosome or the sister chromatid. And we have already read the polyploidy, that is extra chromosomal sets, that is 3n, 4n or either 5n pentaploid. And inversion, which is in inversion a segment is either changed like A, B, C, D, E, F, it is changed to A, B, E, D, C. It means this C, D, E is rotated 180 degree. That is, ये जो C, D, E था वो उल्टा हो गया, ये C किधर चला गया, इधर चला गया. It means जो C, D, E था वो कैसे हो गया, वो ऐसे घूम गया. It means E, D, C, that is 180 degree का rotation हो गया, and this is known as inversion. Now fragile sites are prone to or they are susceptible to breakage, chromosomal breakage. Tetrasomy is 2n+2. सो आई होप आपको हर एक टर्म अब समझ आएगा कि ये जो मेरा मैच द कॉलम फॉर्मेट देने का एम क्या था, कि हम एक एक टॉपिक को पढ़ें और समझें ताकि हमें ये मैच द कॉलम हमें रटना ना पड़े, हमें कॉन्सेप्ट समझ के हम सॉल्व करें. अगर आपको अब भी समझ नहीं आया तो आप जाके उस टॉपिक को फिर से देखिए, वीडियो को प्रीवियस करके, एंड फिर जाके इस मैच द कॉलम को सॉल्व करने की कोशिश कीजिए. अगर आपको वीडियो समझ में आया हो एंड अच्छा लगा हो, सो हिट द लाइक बटन, एंड इफ़ यू वांट मोर, so subscribe the channel, and thank you so much for watching.